Our next speaker is Negin Sheybani. Negin is a PhD student at UW-Madison working with the Peñagaricano lab, where her research focuses on quantitative and statistical genetics using genomics and computational approaches to study complex traits in animal systems. Please welcome Negin.
Thank you, Jimena, for the introduction. Hi, everyone. I'm Negin. And today I talk about our research on the associations between the fecal microbiome and the methane emission traits in Holstein cows. So first I start with why methane emission is something that we need to care about. So methane represents 11% of the total greenhouse emissions and is the second most important greenhouse gas after CO2. Among those methane resources, enteric fermentation accounts for 27% of the total US methane emissions. And enteric methane also can be considered as an energy sink, which means that the energy lost as methane during enteric fermentation can be used for the production and growth of the animals. So reducing the enteric methane emissions not only benefit the environment, but it also can improve the feed conversion efficiency for the productive purposes. Now I talk about the current efforts in this area, and it's the same thing that Francisco, Umit, and Afis talk about in the morning, especially on the genomic solutions. So the big picture of this project is to mitigate enteric methane emissions by combining different solutions. First, genomic solutions. So we want to implement national genetic evaluations for the methane emission traits and update the selection indices, as the changes that we made through the selection are incremental, permanent, and cumulative. And it's a very powerful tool to make the changes in the long term. The second solution is the microbial solutions, and we want to evaluate the rumen microbiome composition and activity and the correlations with the methane production. Also, we want to use the fecal or oral microbiome as the proxy for the methane emission. Third is the spectra-based solutions, and we want to use the milk spectra data to predict the methane production. And lastly, nutritional solutions by evaluating the genotype by diet interactions, cows versus manure emissions, and evaluating the diet digestibility, which my focus is on the microbial solutions and specifically on the using of the fecal microbiome as a proxy for the methane emissions. So now I'll explain briefly how the methane is formed in the ruminants like cattle. So methane is formed through the complex microbial fermentation process in the rumen. During this process, methanogens, which is the type of archaea, form a symbiotic relationship with bacteria. So bacteria utilizes food and produce CO2 and hydrogen as the byproducts of their fermentations, and methanogens use that CO2 and hydrogen and produce methane ultimately. And the important note here is that 95% of the methane produced in cattle is through the lactation, not through the flatulence, as is often assumed by people. So, as I mentioned, 95% of the methane in cattle is released through the lactation. And there have been so many different methods developed to measure the methane production. But one of the most reliable methods is the green feed system that we can see the scheme of this green feed system here. This is the voluntary system at the farm, so cows can go visit the green feed whenever they want. And every time that they visit the green feed, they receive the pellets up to 5-6 times in a day. So they're willing to go visit the green feed and receive their pellets and being recorded by the machine. So this system delivers reliable estimates of enteric methane emissions. And the key here to have the reliable estimates is to obtain numerous short-term records measured at different times of the day for multiple days in a week. And this is what is happening in the farms. So we are running different trials in the farms for about 8 to 10 weeks, and we are collecting the methane productions using the green feed system that I just showed. And at the last week of the trials, we are collecting fecal, buccal, and rumen samples from the cows. And I should mention that we collect the fecal samples from all the cows, but rumen and buccal only from our extreme cows that are in very high or very low and very low in the methane production, which I'll explain some of the results regarding analyzing the fecal samples and what is our goal to do that. So we want to link our fecal microbiome with the methane emissions in cows, but why do we want to do that? What is our goal? First, we want to see what we can learn from the fecal microbiome and the methane production in terms of the biology. Second, can we use the fecal microbiome to predict the methane emissions? And third, can we use the fecal microbiome as the proxy trait in the selective breeding? So to evaluate those hypotheses, to test them, we collect the fecal samples from the cows with the known methane production, as their methane production is captured by the green feed machine. And then we did the DNA extraction using phenolchloroform protocol. We did the 16S RNA gene sequencing targeting both bacteria and archaea. And then we did the data analysis. So here is just an example of how the data looks like after we got the sequencing data and the initial analysis. So we always ended up with the ASV abundance table or amplicon sequence variant abundance table. But this table is basically showing our microbes on the first column and their relative abundances across different samples on the other columns. So in our case, we have 700 samples, so we have 700 columns, and we ended up with 10,074 ASVs or microbes. So we have 10,074 rows, and their relative abundances across different samples. And also we have the taxonomy information of those microbes. So what we did first with that microbiome abundance table, we evaluated their correlations with the methane production. And we found that some archaeas like Methanosphaera and Methanobrevibacter are positively correlated with the methane production, which suggesting their direct role in the methane production. Also, we found that some of the bacteria, like Succinobacterium or Eubacterium coprostanolygenes, are negatively correlated with the methane production, which suggesting that those bacteria probably competing with the methanogens for the hydrogen substances. As I mentioned, methanogens use the CO2 and hydrogen to produce methane. So if there is any bacteria that's competing with them for the hydrogen substances, will have less methane production. Also, after getting the microbiome abundance table, what we did was the enterosignature analysis, and it's the same analysis that Arlan explained earlier today. So by enterosignature analysis, we could find a group of microbes, or it's called enterosignatures, that are co-occur across different samples. So we have around 700 samples, and we found 6 groups of microbes, or 6 enterosignatures, that are co-occur across our samples. And here in this plot, we ranked the animals from the low RMI to high RMI. And RMI is the residual methane intensity, which is the methane production adjusted for the metabolic body weight and milk energy. So we can see that here kind of quite distinct microbial composition between the animals that are low methane emitting cows versus the ones that are high methane emitting cows. And if we focus on 2 enterosignatures, enterosignature 1 and enterosignature 6, as they were significantly correlated with the methane production, we can see that we have high contribution of enterosignature 1 in the high methane producing cows, and we have a higher contribution of enterosignature 6 in the low methane producing cows. And enterosignature 1 was positively correlated with the methane production, and 6 was negatively correlated with the methane production. And then we take a look at their composition of those enterosignatures to see what they are composed of. Here we can see they have quite distinct microbial compositions from each other. And then again, if we focus on the enterosignature 1 and 6, we can see some genera that are specific to those enterosignatures. For example, in enterosignature 1, we have the genera of Alistipes and Bacteroides, whereas in enterosignature 6, again, we have the Eubacterium coprostanolygenous, and monoglobulus that are specific to this enterosignature. And we can conclude that probably on enterosignature 1, we have the genera that their fermentation activities are in favor to the methanogens. And in enterosignature 6, probably those genera that are competing with the methanogens for the hydrogen substances. And also, after getting the microbiome abundance table, we evaluated the heritability of the amplicon sequence variants, or just basically our microbes. And we can see that some of them have near to 0 heritability. But as we go from left to the right of the plot, we can see that the heritability gradually increases, which suggesting that the host can have the control on the abundance of the different ASVs, or abundance of the different microbes. And also we evaluated the genetic correlations between the methane production and all the ASVs, but in this plot we have the genetic correlations between the methane production and top 5% most heritable ASVs or microbes, and we can see that some of them has near 0 genetic correlation, some positive, and some negative genetic correlations. So as a take-home messages, fecal microbiome shows a promise as a non-invasive proxy for the methane emission traits in dairy cattle, and it's easy to use, cheap, and non-invasive. Also, distinct microbial communities can explain why some cows are emitting more methane than the other ones. And also, a large proportion of the fecal ASVs are heritable, which suggesting that the host can have the control on the abundance of the different microbes or ASVs. And as the future direction, keep collecting more data, more samples, and keep expanding our data set. With that, I'd like to thank my advisor, Dr. Francisco Peñagaricano, and our lab group, also Dr. Hilario Mantovani and the lab group for the collaboration. And a special thanks to all the researchers and technicians that go to the farm all the time and take the samples and data and make this research possible. And also a special thanks to all the industry partners, especially their Innovation Hub, for their support. Thank you.
All right, we have a couple minutes for questions, so the floor is open for Negin.
Great talk, Negin. So I was curious about the archaea in the fecal samples. Did you find any enterotypes that have this signature, or have you been able to evaluate them in a more precise way?
Good question. When we did the enterosignature analysis, I didn't find any specific archaeas, probably because their abundance is lower than the other bacteria in the samples, and that's probably, I think, why we didn't see them. But when I did the specific correlation between the microbes and the methane production, I could found them and I could see them. Yeah.
One quick last question, anyone? All right. If not, let's thank Negin.